Welcome everyone. Imagine walking into your kitchen in the year 2030 and seeing a small desktop machine next to your coffee maker. But this device is not for brewing drinks. It is for editing genes. What once required a full laboratory can now be done in a space no bigger than a toaster. This is not a science fiction scenario. It is the emerging world of decentralized genetic engineering powered by CRISPR technology. CRISPR has already transformed laboratory biology. Now it is stepping into homes. That shift raises important questions about access, safety, and the future of human capability. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It is a method originally derived from bacterial defense systems. In nature, bacteria use CRISPR to recognize and disable invading viruses. Scientists figured out how to harness this system to target and cut DNA sequences with extraordinary precision. The key components are the CA's enzymes, which act like molecular scissors, and a guide RNA that tells the enzyme exactly where to cut. By swapping out the guide RNA, researchers can program the system to modify nearly any gene in any organism. Since its discovery in 1993, CRISPR has revolutionized biotechnology. It has been used to treat rare genetic diseases in clinical trials. It has edited the genes of crops to improve yields and resist pests. It has helped create animal models of human diseases for research. But all of this has so far required trained personnel and highly controlled environments. That is now changing. With the rise of open-source biology and biohacker communities, CRISPR kits are being developed for non-experts. And by 2030, such kits may be as common as a 3D printer or soldering iron. A decentralized genetic engineering kit typically contains a set of reagents such as CRISPR enzymes, plasmids, bacteria, and DNA templates. It also includes instructions for growing bacteria and inserting DNA. Many kits come with visual indicators like color-changing bacteria to confirm whether the modification worked. While these tools may sound advanced, they are increasingly affordable. Some basic kits already exist today for under $200. They are sold for educational use, but many users go far beyond the classroom. The barriers to entry are rapidly falling. In this new biohacking world, enthusiasts are using CRISPR kits to create glowing plants, customize yeast strains, or engineer bacteria that break down plastic. These are small-scale experiments, but they reflect a growing movement of people who want to explore biology as a creative medium. Online forums document successes and failures. Video tutorials walk newcomers through the basics, and distributed labs offer physical spaces for shared experimentation. By 2030, this network could rival the early days of home computing in its energy and innovation. There are also commercial efforts aiming to streamline this trend. Companies are building plug-and-play systems where users can design genetic sequences through a software interface. The system then prints the guide RNA, mixes the components, and executes the gene edit in a sealed chamber. This approach combines automation with safety. No one needs to handle raw DNA. Instead, users design a genetic change the same way they might design a three-dimensional object for printing. As the cost of DNA synthesis drops and automation improves, these systems could become household appliances. But with this power comes real risk. Gene editing is not like writing code. Living organisms can mutate, spread, and interact with the environment in complex ways. An accidental release of a modified bacterium could have unpredictable consequences. There is also the risk of intentional misuse. While it is unlikely someone could make a deadly virus with a basic home kit, the line between curiosity and danger is thin. Many countries have vague or outdated laws when it comes to DIY biology. That is why safety guidelines and community norms are so important. Leading biohacker groups have developed codes of conduct and safety checklists. They encourage containment measures like using kill switches in modified organisms. Some community labs require training before anyone can use CRISPR tools. Others maintain access logs and monitor projects. This decentralized governance is essential in a field where official oversight may be slow or ineffective. Meanwhile, education is playing a major role. Universities are partnering with makerspaces to offer gene editing courses. High school biology classes are integrating CRISPR experiments into their curriculum. Science museums are hosting interactive exhibits where visitors can simulate gene editing on touchscreens. The idea is not just to teach the science, but to foster a culture of ethical curiosity. If people understand how gene editing works, they are more likely to respect its power and use it wisely. Another area of growth is personalized medicine. In the future, individuals may use home gene editing kits to tweak their own microbiome. Instead of taking probiotics from a bottle, users might grow their own engineered bacteria designed to aid digestion or produce vitamins. Some researchers imagine kits that allow for screening genetic traits at home. Combined with wearable health monitors, these tools could support highly personalized health routines. There are still many technical and ethical hurdles, but the foundation is forming. Animal enthusiasts are also joining the movement. Some biohackers are experimenting with gene editing in model organisms like fruit flies or zebrafish. There are attempts to create designer pets with traits like bioluminescence or enhanced coloration. While this raises animal welfare concerns, it also shows how accessible the tools are becoming. Breeders might one day use CRISPR to eliminate hereditary diseases in dogs or cats. As the technology matures, ethical guidelines will need to keep pace. In agriculture, small-scale farmers might use CRISPR kits to modify crops for local conditions. This could include drought resistance, pest deterrence, or faster growth. Instead of buying patented seeds from large corporations, growers could customize seeds themselves. This decentralizes agricultural innovation and may improve food security in remote areas. However, it also raises questions about regulation. Modified crops must be tested for safety and environmental impact. A balance will need to be struck between empowerment and oversight. Looking ahead, the rise of at-home gene editing could spark new industries. We might see the emergence of biodesign as a profession. Just as graphic designers work with pixels, biodesigners could work with DNA. Their creations could include custom bacteria, engineered fibers, living art, or therapeutic compounds. Online marketplaces might emerge where genetic designs are traded like software plugins. Verification and safety would be crucial, but the creative potential is immense. There is also the potential for citizen science. With more people able to perform experiments, the collective data pool expands. Networks of amateur researchers could help identify new antibiotics, monitor environmental changes, or study local biodiversity. Platforms might emerge to coordinate projects, share results, and connect experts with hobbyists. In the best case, this democratizes science and accelerates discovery. In the worst case, it leads to reckless or intentionally destructive experimentation. Finally, we must consider the cultural implications. What does it mean when individuals can rewrite the code of life from their homes? Will gene editing become a normal hobby like gardening or coding? Will there be status symbols based on engineered pets or personalized microbes? As we enter the next decade, the tools of biology are becoming tools of everyday creativity. CRISPR is no longer just for professionals in lab coats. It is for artists, hackers, students, and anyone curious enough to explore. Like the early days of personal computing, this moment carries both promise and peril. The future of biology will be written not just in research labs, but in kitchens and classrooms around the world. Thank you for watching. Please like and subscribe.